Hello and welcome to the 25th Penprig video. About 6 months ago, with tongues somewhat in cheek, we challenged the Virtual Biotech team to speak to us about their work in plain English. They met the challenge in style in 3 videos that are now available to all comers in the Penprig YouTube library. We have invited them back today for a question and answer session with a panel that includes members from 3 research interest groups around the UK. So now I'd like to introduce Arthur Roach, the Director of Research at Parkinson's UK.
So hello to the Peninsular Parkinson's Research Interest Group. Really glad to have the chance to talk to you. Great to have your questions. I can tell that you have great interest in this program because of the very thoughtful questions that have been suggested. And I want to, give some comments on a few of those. One question was about the value for money, the efficiency of the work we do, the balance of the priorities to discover increased understanding about Parkinson's, balanced by perhaps the profit motive. So on that, a couple of points I'd make. One is that the purpose of the Virtual Biotech program is not to increase the understanding of Parkinson's. It's the kind of research that does that, genetic research, biochemical research. We do fund that in Parkinson's UK through other programs, through research grants, usually to university investigators. Many other investigators, Medical Research Council, ASAP, also fund that kind of work. Absolutely essential. We fund it, other people fund it. Then the Virtual Biotech program is a little bit different. We start from that understanding and say, okay, here's a very compelling hypothesis about a way to make a new treatment for Parkinson's. Let's focus now on creating that new treatment. Not the understanding is already there. We're going to try to create a new treatment. That part of work, in fact, there's not a lot of conflict between our objectives, creating a new treatment, and the profit motive of the biotech companies, the pharma companies, and the other investors that also support this kind of work, perhaps alongside us. To create a new treatment, it's going to require a lot of funding, a lot of investment. Pharma companies will have to get involved. Other investors will probably have to be involved. And so our objectives, our priorities are very much aligned with theirs. Let's get success. Let's find a drug that will really work. Let's get it to patients. So there are relatively few actually conflicts between their priorities and ours. On the question of general efficiency and effectiveness. So to do this kind of work, this creating the new medicines, testing them, safety testing, pharmacology testing, manufacturing, all these things, very critical for a new drug. To do those things, yes, they could be done by different partners. They could be done in university by the investigators there. They could be done by a biotech company with dedicated management, or they could be done by a contract research organization. So we at Parkinson's UK could design a research plan, and we could contract with a laboratory that does chemistry work or biology work to do the steps, and they'd be sort of executing our research plan. All those 3 can work, but the people running this program, myself and others, have a lot of experience with this. Many times we've been the ones on the other side of the funding as well, and we really firmly believe that in most cases, the most efficient, the most effective way to deploy any funding is actually with the small biotech companies. They have a real focus. Sometimes it's the only project they have. They have a small number of people who are experienced in the business. They know how to do it. They don't spend extra money on buildings and overheads and staff and logos. They're very lean and focused. The people in the company know that a successful project will mean their successful career. So there's a dedication and a level of intense thought and effort that we really don't get from either of the other possible executors of the plan. So we work with the biotechs, not because we like to work with biotechs. We do it because we truly believe this is the most effective way to generate high quality new drug candidates. So that's a part of our philosophy. There was another question about whether any really important new discoveries have been made through the Virtual Biotech funding that would not have been made otherwise. So yes, there are. Certainly, the clinical trial you may have heard about, Nerlixis 112, tested for dyskinesia and Parkinson's. That drug would never have been tested in Parkinson's without Parkinson's UK Virtual Biotech support. It wasn't going to happen. The company had been looking for funding. They didn't get it. So we came in, and it led to eventually Michael J. Fox joining and co-funding a stage, and a positive clinical result showing that molecule really does reduce dyskinesia in people with Parkinson's. So that could go forward now with other funding to become a drug in regular use. Very important discovery. Another one I'll cite is our NRG company. So this is one that, again, had no funding. They've been looking for funding. We were the first ones to fund them. We've funded them over 3 years now. Other investors now have come on board. They have made a discovery about a mechanism of action that could be neuroprotective in Parkinson's. I can't provide details because they haven't published it yet, but they will publish it. And this publication will show that a protein of previously uncertain function actually is definitely involved in this neuroprotective mechanism that they're studying as part of the mitochondrial involvement in Parkinson's. And so that's a discovery they made with funding from us, and that's going to be published, and the whole world will be able to potentially benefit from and improve on that. So definitely discoveries are being made, in addition to every other project is making incremental project that's not as dramatic in the individual case. The last comment I'll just make, a very general comment, is that to succeed in this business, there are a lot of things that are necessary. Everything has to work. There can't be any pieces that don't work when you're trying to make a new medicine. But 3 very important pieces that we rely on are, number 1, patient involvement of people like yourselves, talking to us, giving us your thoughts on priorities, giving us your thoughts on what clinical trials could work better, design of studies, that sort of thing. Very essential. Probably Parkinson's UK is the one player in this space that does more of this and takes it more seriously than anyone else. So thanks for working with us. Secondly, we need people to come forward and participate in clinical trials, even guinea pigs. This is not for everyone, of course, and all trials are different, but many people who come into trials really find it a good experience. They get more time with the doctors and nurses, more time in the clinic, and know they're doing something very important. And also they get early access to an experimental treatment that might help them that might not otherwise be available to them. And thirdly, of course, money. For us to do our role, for us to influence the biotechs and the pharma companies and the other investors, we have to be able to carry our end of the stick. So any funding support you provide Parkinson's UK to allow us to operate the Virtual Biotech program is absolutely essential. Without it, we wouldn't have this voice in the business of creating new treatments for Parkinson's. So thanks a lot. I know the other people in my team have enjoyed interacting with you, and I hope someday I'll have a chance to meet you in person as well. Thanks. Bye.
Our speakers today are Associate Director for Research Communication, Claire Bale, Drug Discovery Manager, Jan Kulagowski, Clinical Studies Portfolio Manager, Shona Clegg. And our question master for today, Liz Nash, Research Network Support Lead. East Midlands Research Interest Group, we have John Turner, Peter Barrett and Martin Rumsby. From the west of Scotland, we have Sue Keast. And from Penprig, we have Nigel Coleman. So without further ado, I will hand over to Liz Nash and get the party started.
Thank you, Chris, and thank you, Penprig, for inviting us to be here today to answer all of your questions about our Virtual Biotech portfolio of work. Thank you to everyone here today that has submitted questions, and I believe Arthur Roach, our director of the Parkinson's Virtual Biotech, has already answered a couple of the questions submitted by the East Midlands Research Interest Group. But I just wanted to ask Peter, Martin, and John from the group if you had any follow-up questions arising from Arthur's answers to your questions. I know he's not able to be here live today, but team members might be able to jump in. Martin.
Yes, I do. Thank you, Liz. I would have liked to ask Dr. Roach at what stage Parkinson's UK is at in getting the exciting new drug NLX-112 into a bigger trial to explore its effects on progression mediated perhaps by GDNF. As far as I can make out, Neuralixis UK doesn't have any laboratories, staff or money of its own. So I wonder how you're going to go about getting money to implement a bigger trial and who's going to do the work.
Thanks, Martin. I'll start, but Shona might know more as well and might want to chip in. So Neuralixis are actually, I believe they're kind of a French-American company, and rather than based in the UK. And they are, the last time we spoke to Adrian, who is I think the chief executive of Neuralixis, he was in the process of starting to talk to other potential investors who are very interested in providing funding to Neuralixis to help them take it into the next phase of trials, which will be phase 2B. So this phase of trials is likely to need quite a lot more money than we initially invested, so probably in the tens of millions of pounds is my understanding. But based on the really positive data that came out of that phase 2A that we co-funded with Michael J. Fox, he's very optimistic that they'll be able to secure the funding they need to get that going fairly quickly. And it's, you know, something we'll be following very closely. We want to stay involved and keep abreast of what's going on with Neuralixis, and also have the ability to share that with the Parkinson's community. So we will definitely be asking for an update from Neuralixis and Adrian before the end of the year, and letting people know where it's got to so far.
Thank you very much.
Thanks, Claire. And Peter from the East Midlands Frig, you have a follow-up question.
Well, I do. I think you're quite right. And thanks very much to everybody in Pembroke for asking us along. It's a pleasure to be with you. The question was answered by Arthur Roach, of course, in the first question about how do we know it's delivering good value for money. One of the follow-up questions would be, how often is it scrutinised? How many of the projects are scrutinised? And who does the scrutinising? And who pulls the plug if it's not working? Sorry, there's 3 questions.
I don't know, Jan, you might be best placed to talk about the preclinical side on that front.
Sure. So every project has a team of people called the Joint Steering Committee that oversee the project. They monitor progress, they provide advice, and the JSC would be comprised of people from Parkinson's UK. So I sit on a couple of those for the projects I have responsibility for. There may be external people, such as specialist consultants if required. There'll be representation from the project owners, so it might be a small biotech or an academic group, as well as the CRO. As Arthur explained, we tend to work with CROs, so contract research service providers. So it's quite a multifactorial team, and we on the projects I'm involved in, we meet every fortnight and get an update on the project, discuss results, plan the next studies, and we review the project. So when we assign funding for a project, let's say we agree to provide £1 million for a year to a project, and that was quite standard for us. That money wouldn't be handed over in one go. It would be split into 2 or 3 chunks, and progression from one tranche to the other would require achievement of milestones that would be set at the beginning. So the project would be monitored, and we'll see how well the milestones have been achieved, or to what extent, and the project will then be presented to an external panel of experts that we call upon, and they will scrutinize the science. They're all very experienced people with 20 or 30 years industrial experience, and recruited from different disciplines relative to drug discovery, so chemistry, biology, pharmacology. And in fact, we had one such meeting just the other week, where we reviewed a couple of ongoing projects, as well as some new ones that they gave their view on. So there are many checks and balances along the way. We don't provide our money, easily. We have ways of making sure that the money is spent appropriately and transparently as well.
Thank you.
Thanks. Shona, you might want to say something about clinical too.
Yeah, I was going to say on the clinical trial side, it's slightly different. because there are some ethical considerations in terms of starting people on a clinical trial. So once a trial's up and running, the payments that we schedule out at the start are usually, again, like the preclinical, milestone based. So they might be first patient enrolled, they'd get a portion of the funding. Last patient enrolled might be another trigger point. But also, as we progress through the trial, we have regular meetings with them. So not quite as regular as the preclinical, because the trial is a plan that's already fully laid out, and we don't have to consider what, as Jan said, what the next experiment's going to be. We know everything that's happening. It's all been planned out in advance. So we join the study sponsor for regular updates on where they're at with recruitment, timelines, whether they're having any additional difficulties with the trial, and also whether there's anything that we can support them with. So things like communications around recruitment, updates to the trial participants, any working groups, or studies that they may want a focus group that we can help support set up.
Thank you, Shona. John.
I was going to ask, there's a lot of different research going on, and one of the aims of the Virtual Biotech, as I understand it, is to look at various different possibilities. One thing that I was wondering was, does that include non-drug attempts to deal with things like dyskinesia? I'm thinking of there's some work being done at the moment on using the vagus nerve or the median nerve to try and prevent tremor and dyskinesia and so forth. And would that feed into the Virtual Biotech, or would that be treated as separate?
Thanks, Don. That's a really good question. So the Virtual Biotech is specifically set up to look at drug treatments for Parkinson's. But we do, as I think Arthur mentioned in his video, we have a whole other set of ways that we fund research, our research grants program. And we have a dedicated grant scheme, non-drug approaches, where we're exactly for that kind of intervention. So we're actually funding a study looking at using a stimulation device to stimulate, I think it is the vagus nerve, to look at if that can improve movement for people with Parkinson's. We've also funded studies looking at things like physiotherapy techniques, cueing, all sorts of different ways. There's one looking at different approaches we can use in physiotherapy to avoid freezing, for example, alongside things that can help with mental health, such as talking therapies. So there's, we know how important that is for people with Parkinson's, having those other ways to manage symptoms, and also recognizing that some of these kinds of research projects could deliver something more quickly, because it's maybe a simpler intervention than something like a drug, which we know takes many years to be approved and made available on the NHS.
Thank you.
Thank you, John, for your question, and Claire for your answer. I'm now going to come to Sue Keast from our West of Scotland Research Interest Group for a question from north of the border.
Good afternoon, and thank you very much for inviting me along, Chris. I really appreciate it. At the West Scotland Research Interest Group, our question was concerning genetic research, and we're just interested in learning more about what research is being carried out on the genetic form of Parkinson's, and perhaps what a cure for that genetic form might look like. Thank you.
Hi, Sue, and thank you for your question. So the genetics of Parkinson's are very, very complicated, and I don't think we have a full understanding yet of, the full picture of all the genetics that might be involved. There are very few people who have a form of Parkinson's which is directly and solely caused by genetics. I think the most definitive kind of genetic form of Parkinson's is one with where families inherit a change in the gene for alpha-synuclein, which I'm sure people on this call are familiar with. This is the gene that makes the protein alpha-synuclein, which we know misfolds in Parkinson's and leads to the development of things like Lewy bodies. And people with that particular mutation produce too much alpha-synuclein, and that means that they are pretty much, I think, guaranteed to develop Parkinson's, and they usually develop it at a very early age. But that is very unusual. I think, you know, there's only a handful of families that have that particular genetic mutation. For the most part, the genetics that we know about are more to do with increasing risk of Parkinson's. And the two most common genetic factors that we know about so far are LRRK2, LRRK2, and GBA, so mutations in two different genes. And there is a huge amount now of research into developing treatments that target the LRRK2 protein, which is made by the LRRK2 gene, and looking at the GBA protein, glucose cerebrosidase. It's an enzyme that's made by the GBA gene. So the understanding of the genetics has driven this, you know, huge amount of research into specifically those things. And that's true of the more minor genes as well, to a lesser extent. And there's also a huge amount of research going on into alpha-synuclein. So while there's lots of research going into these sort of these proteins, drugs built on these proteins that we know about through a lot of the genetic research, we hope that these treatments that are being developed might be helpful for all people with Parkinson's as well, because we know that it's common. Alpha-synuclein misfolding and accumulation is common across many people with Parkinson's, as is problems with waste recycling, which is what GBA does. So hopefully the solutions to the genetic forms will also provide solutions to the non-genetic forms. And actually in the Virtual Biotech program, we are co-funding a clinical trial of Ambroxol, the cough medicine some people may have heard of. It's a constituent of cough medicine. And specifically Ambroxol, we think, acts through GBA, so glucose cerebrosidase. So we're in that trial, they're looking for people who have that gene mutation as well as people who don't, to see whether it's more potent potentially in people with the mutation, but hopefully it will be useful for all people with Parkinson's. It's a long answer, sorry about that, but hopefully useful.
Thank you, Claire. Long answers welcome.
Thank you.
I have a question now from a member of the Parkinson's community who watched the videos produced by the Virtual Biotech team. And this person asks anonymously, they would like to know more about the molecules that you referred to throughout the Virtual Biotech talks. So, you know, how does a biotech start this process of looking for molecules? How do they identify ones that have potential to go on to be drug treatments, especially ones that we know or hope might cross the blood-brain barrier? So I don't know, Jan, if you might like to take a stab at that first, please.
Sure, thank you. Yes, great question, and one that I spend many years, or have spent many years worrying about myself as a chemist. So where do the starting points come from for drug discovery projects? Well, you have to have a mechanism, a pathway that you believe in that could be interfered with, if you like, to cause or provide a beneficial effect. And with that in hand, that there are two major ways of finding starting points, chemical starting points. One is to see what other people have done, which sounds perhaps a bit strange, but that's a very common way of doing it, and we give it quite a fancy name rather than sort of just taking ideas from other people. We call it a knowledge-based approach. So we would look in the literature to see what's known, what people may have published. So these could be academic groups who have done some work in the past and maybe found some molecules which act on that pathway of interest. Also, we would look at what other companies have done. So we'd look at their patents. So companies tend to be fairly circumspect. They don't tend to say very much about what they're doing, because what they're doing has a lot of commercial value. So there's a lot of confidentiality associated with it. But to protect their assets, they would file patents, and in those patents would be many molecules, and then the trick would be to try and work out which ones are the important ones. With that in hand, we would then start to change those molecules to make them better for our requirements. So it's very likely that other people may have been working on that pathway for a completely different condition than Parkinson's. So many of the, or some of the projects that we have on board, they're specifically sort of targeted at Parkinson's as far as we're concerned, but that same pathway could be useful in cancer or other conditions. And so the molecules for those other uses perhaps wouldn't be appropriate for Parkinson's, where we would have to get the molecule into the brain, which is a very difficult process because the brain really doesn't like foreign matter to enter it, and it has an exquisite array of methods for stopping that happening. So we have to try and fool the brain, or to try and get material into it in order to exert its positive effects. So there's a lot of work to be done, even if you find something that seems to be of interest in literature. And also the other important aspect is it has to be new and novel if it's going to have any commercial value. And whilst we don't work to make a profit, we're a charity, our main objective is to help people with Parkinson's in any way we can. But in order to attract third-party investment later on to take on the project, where it becomes much more expensive, as we heard with the Neuralixis program, the next phase of clinical studies will be much more expensive perhaps than we would be able to cover. And so companies are there to make a profit, and so there needs to be a commercial aspect to it. The other way of finding new molecules is to look randomly or semi-randomly, and that's normally through what's called a high throughput screen. So you'll take a library of molecules, and pharma companies have libraries of molecules that they've made over the years or bought in, and those could number millions of molecules. And they would have a biological test that's automated, and they would screen all those thousands or tens of thousands of molecules and see which ones may have activity. And then you start working through optimizing, making those molecules better, because anything that comes out of that sort of screening process will perhaps only have biochemical activity against the target, but won't the target protein or pathway, but won't have the ability to get into the brain or resist destruction by the body through the liver or whatever, which is another major issue that we need to consider. So I don't know if that's answered your question. I could go on for ages, so I better stop now, because it's one of my pet subjects. So I don't wish to bore you too much.
Well, thanks very much, Ian. It's fascinating, and it's prompted Chris to think of a follow-up question. So Chris, I'll come to you, please.
It's more to do with being polite. It was most remiss of me not to introduce David Bousfield, our privateer, I suppose we would refer to him.
Thank you, Chris, and welcome, David. Ian, thank you for your thorough answer to the last question, and taking a slight turn away from molecules, perhaps, a question which in my mind is somewhat related, also submitted anonymously by a member of the community. So this person has asked, with biotech companies and academic partnerships driving forward and leading Parkinson's research, does any aspect of the Parkinson's UK approach towards research funding consider any alternative therapies or homeopathic medicine for research investigation? And are these types of treatments something you might explore within the Virtual Biotech portfolio or elsewhere?
So I think this kind of follows on from the question we had earlier about non-drug therapies. So as I mentioned then, we have our non-drug approaches grants round. So I think this year we're funding a further 3 or 4 new projects in this space. So we are really open to exploring non-drug approaches alongside all the work that's going on in the Virtual Biotech program. Things like alternative, like homeopathy and other alternative medicines. I think it's really important to know that these are things that people have the ability to try for themselves and, use out alongside the medicines that they get prescribed by their specialists. They may not need to have the same level of evidence. And certainly, I mean, things like homeopathy, there isn't really any evidence, but many people like to try and take things that, they think will help them. And there is evidence that taking placebos helps people. So, it's important that we try and pick the things that we think there's a real likelihood that they can help people with Parkinson's, and develop the research so that it gives the evidence that's required either for people with the condition to feel confident to maybe try these things themselves, whether that's an exercise-based therapy or something to do with diet, for example, or to build enough evidence for things like physiotherapy practices that they can start to be implemented within the NHS. But it's a different kind of research to that taken forward by the Virtual Biotech, which is very much a pharmaceutical based approach. So we're looking to do really, ultimately big, large-scale clinical trials to prove the effectiveness of drugs that can then be approved, adopted, and distributed to people with Parkinson's. And the level of evidence there is really, really significant. So I think that's the difference, and why there needs to be different approaches to those two kind of strands of work to develop better therapies.
Thank you, Claire. I think that's really clear. And you mentioned diet in your answer, and that leads nicely onto our next question, also submitted anonymously from a member of the community. So this person asks that the proper scientific research of diet and food additives is largely sort of undiscovered territory or country. Do you perceive a role for a virtual biotech, or more generally for any Parkinson's UK funding stream, to sponsor this type of academic research? And apologies, this question was from the PenPrig group.
Yeah, so yes, I think we would be open to funding research into diet-based potential therapies. We have in fact co-funded or part-funded a study looking at a probiotic drink for people with Parkinson's, and those results are due out very soon. We've also funded more early stage research looking again at the sort of the makeup of the gut microbiome in people with Parkinson's, and looking to see what is the imbalance there, if any. Is it different to someone without Parkinson's? What can we do to foster a healthy gut environment and potentially improve symptoms, and possibly even, I mean, research in this space is still at an early stage, possibly even slow down the course of Parkinson's. Some people and scientists are starting to see that potentially Parkinson's may in some people start in the gut and be accelerated through things that are happening in the gut. There's also been, while we haven't funded it, we do take a really keen interest in understanding what's happening globally in this kind of area so that we can provide good information to the Parkinson's community. So there has been research looking at various different kinds of diet in people with Parkinson's. And I think from everything that I've read and listened to over the last few years, it seems like the Mediterranean diet is the leading diet, not just for Parkinson's really, but for health in general. It seems to tick a lot of the boxes, and so that seems to be the thing that's recommended. And yeah, I mean, there needs to be more done, but diet research is very, very tricky to do because there's so many variables. How do you do a clinical trial of diet where people, different cultures and just different preferences, people have very wildly different diets, and most people eat different things every day rather than eating the same thing. So it's a very difficult kind of science to do well and definitively, which is why maybe there hasn't been as much progress in this space as many people would like to see.
Thanks very much, Claire. I'd like to come to Shona now for a question also submitted by the PenPrig group. And they've asked, what influence do you and the Virtual Biotech team have over the development of patient or participant-facing documentation for the clinical trials that you sponsor? And what initiatives are already in place or could be put into place to improve the involvement of people with Parkinson's in shaping clinical trials?
Okay, so firstly, I should probably highlight that the Parkinson's Virtual Biotech doesn't sponsor any clinical trials. We fund or we co-fund them. It sounds like a really trivial differentiation, but it's a really important one because the trial sponsor is legally responsible for the setup, running and reporting on the trial, and that's not our role. But having said that, as the funder, we can have quite a significant input on the documentation that goes out, the review and that side of things. So a really good example of that is the Top Hat trial that's currently recruiting. It's a trial of ondansetron as a treatment for Parkinson's hallucinations. And what they did was they worked with Parkinson's UK to set up a focus group made up of people with Parkinson's and people with experience of caring for family members with Parkinson's hallucinations. And they were involved in the very early stages of the study planning. They made important contributions to things like the study design, including the timing and location of visits, as well as inputting into that documentation that you described. So things like patient and caregiver information sheets. So that was an area where we were able to really influence the documentation of the trial and really get the input from people with Parkinson's into it. So some of the examples were that the focus group pointed out that really lengthy telephone conversations were actually really fatiguing. And so as part of the feedback from that, the trial was designed so that the number of questions asked over the telephone were reduced. And the location of visits in terms of whether they were home-based visits or whether people had to come into the clinic. And another example of documentation tweaks that were done was that the feedback was it was really, really useful to have a one-page flow chart. This is the kind of thing I love, which allows people to see at a glance what's involved in taking part in the trial. And so because of the feedback from this focus group, that was included in the trial. Another area that we have done a lot of work around is what we call our Keeping in Touch toolkit. So this is a set of documentation that's available on our website so that researchers can keep in touch with people and participants on their trial. It's got a series of templates for different stages within the trial so that they can use these templates, make them their own, but it gives them, key timelines to send things out. So, updates when the final subject's been recruited into the study, and key information that people who are part of the study want to know and hear about. So future plans for the research, that kind of thing. One of the other things I should probably point out is this work was co-produced. So it was done between researchers, the Parkinson's community, and also developed with the HRA, which is the Health Research Authority, and the Research Ethics Committees to reflect best practice. So all that information is now in one place and the researchers can use that. And it includes the key feedback that people in the community want to see from the clinical trial.
Thanks very much, Shona. I realize we're rapidly coming towards the end of our question and answer session today for our Virtual Biotech team, but I wanted to ask particularly David, who joined the call and didn't get a chance to add any questions at the beginning, if there would be anything you'd like to submit to the team before we close.
Well, thanks for inviting me, Chris. I'm afraid I think I've absorbed more information than I'm able to give back. It's a long time since I sat in on a Parkinson's meeting like this. Kind of moved on to other areas. I suppose one question I have, which probably should have been asked at the very beginning, a lot of our theorizing about Parkinson's is based on a kind of lock and key notion of how the brain works. Indeed, any living system. So you have things that are working and things that can influence the working of those molecules. And if you believe that the molecule is overexpressed or underexpressed in a particular disease, then you've got a target. You've got a way of maybe producing a drug which, depending on the way the disease takes this molecule up or down, compensates in some way for that. But it's a very simplistic way of thinking about brain function. And I just wonder how, whether through the biotech program or other programs that Parkinson UK funds, you could address the complexity really of disease and what this brings to the table in terms of coming up with new drugs.
That's a killer question, David.
That's
Good
why
to
I
say
didn't ask it until the end.
Yeah, I think it's a really good, an excellent point. And we don't, it goes to the heart of the question of, we don't really know how and why Parkinson's happens. And we have some clues, some of those proteins that go up or down or, misbehave. And a lot of the drug development is based on trying to fix those proteins. But we still need to keep trying to understand better what's going on inside the brain, how that chain of events happens. If some things go up and other things go down, maybe is that cause or effect? And we will be announcing some new research soon. That's going to be using our brain bank and new cutting edge techniques, genetic analysis, proteomic analysis, to generate some of the answers, hopefully, to those questions about, building that picture. So we have a much better understanding of why and how brain cells in Parkinson's stop working properly on a cell specific basis. The advances that have happened in analytical techniques and also data processing means that we'll be able to look at brain tissue in more detail than ever before and hopefully understand better than ever before. But yeah, it's an ongoing mission, and I think that there's other organisations we're collaborating with to try and understand that too. So, in his introduction, Arthur mentioned the ASAP, which some people may have heard of, is Aligning Science Across Parkinson's. It's a global collaborative endeavour, bringing together researchers from right across the world to try and answer some of those questions. And again, we've got to work together to figure those things out, and hopefully come up with better ways to figure out what the new drug treatments need to look like and look beyond just turning proteins up or down. But it's a great question. I don't know if Jan wants to comment further on that.
Have you come across sorry, I'm losing my voice. Have you come across the consortium for the genetics of Parkinson's? It's a multinational program. I think it's partly funded by
so there's a few. I think there's one called the IPDGC or something. So that's been around for a long time, and yes, we are aware of it, but I think it's maybe been slightly superseded by the Aligning Science Across Parkinson's project, which is also very closely aligned with Michael J. Fox Foundation. And they're doing, it's got a global genetics collaboration through that project, where they're not only they're looking at genetics right across different populations. So they've done a lot of work looking at genetics in Black populations, for example, which was a major missing piece in our understanding of Parkinson's. And they've identified that there are some different genetics going on in that population of people. So there's still so many gaps in our understanding that we need to fill, and we can only fill them by collaboration.
Okay, thanks for that.
Thank you, Claire. And always so positive to hear about international collaborations. And speaking of collaborations, thank you, everyone, our research interest groups, for watching the excellent series of videos about our virtual biotech that the PenPrig group put together, and for submitting such thoughtful questions. And that just leaves me to hand back to Chris.
And thank you to everybody for participating today, especially the professionals. It's a great privilege on behalf of PenPrig to represent professionals in the community forums. You don't normally face the camera, and we appreciate that fact. And your research into disease modifiers from Parkinson's is essential. We look forward to more of the same in the future. Thank you.